Alrighty, so in this series, we're going to move into the realm of population genetics, taking a look at microevolution. So in the lectures before, I had talked about macroevolution as a driver for evolutionary change. And macroevolution deals with these large-scale, long-term changes within populations, and these changes occur at or above the species level. Right? So it's closely tied with speciation as far as the creation of new species. Deviating a bit from that, we're going to talk in this series about microevolution. Microevolution is another driver of evolutionary change, but it's a bit different. So micro operates on a much smaller scale. It operates below the species level. So at the level of what you'd refer to as the individual or the population. So this would deal with not the formation of new species, though it can eventually lead up to that, but rather immediately dealing with the nuances that you would see in the genetics of a population, or the creation of subspecies, or what some individuals call rarities, not rarities, I'm sorry, races in a population. So again, we're going to look at that component, kind of take a more narrowed look at evolutionary change. I do have to apologize. Figaro is sitting on my lap, so if you do hear purring in the background, that is just him. And I apologize if it causes any disruption whatsoever. But moving on, so with microevolution, a lot of these changes that we will be talking about deal with the changes in genetics, either the genetics of an individual or of a population. So before we move in, it's good to go over some of these important terms that will come up in the lecture series. Make sure that you are on the same page or same understanding as I am, but also in case you haven't encountered these terms before or haven't seen them for many years, you can kind of get yourself up to speed. So first point, what is a population, right? So as I was saying, microevolution operates at the individual or the population level, right? So in this realm here, above that, that's above the species level, right? That would be macroevolution. So what is a population? I consider a population as being, and what you'll find many biologists defining a population as, being a localized group of individuals that are capable of interbreeding and producing fertile offspring. And that these groups are more or less isolated from other groups or other populations, right? So they're these clusters of individuals, again, that can interbreed with one another, but they can't do quite so much of that activity with other groups, or they're more or less, again, isolated. So those are populations. Gene pool, right? So a gene pool, all populations have a gene pool. A gene pool represents or is comprised of all the alleles that are present within a population at any given time in space or history. So depending on allelic frequencies, or the genetic composition of those individuals, you will influence the composition of a gene pool. Now, typically in conservation biology, right, we're looking for gene pools that have high amounts of variation or diversity within them. Typically, the more diverse the gene pool, the more likely that population will be able to adapt to stochastic change or events in the environment. Because again, you think of variation as backup plans, right? The more variation or the more backup plans that a population has, then the more likely that there will be one or more individuals that can survive some sort of change in the environment. So all of this deals with alleles, right? And in case you forgot, alleles are alternate forms of a gene, right? So you have a gene, say we have gene A. There's going to be two or more forms of that gene. Typically, you have two forms of a gene. We'll keep it simple. So this A gene has a dominant allele and a recessive allele, right? So the dominant allele will be big A. The recessive allele will be little a. So they're the two alleles, right? Big A, little a, both come from the same gene, gene A, but they're different forms of that gene. So they produce a different phenotype. Phenotype are the observable traits or characteristics of an individual. Now, when we talk about, again, the gene pool and these changes in frequencies, right? When we talk about allele frequency, this represents the proportion of a specific allele in a population, right? So that can change within time, depending on the natural variation of genetic composition of individuals. It can also depend on, as we'll talk about in a moment, some other external factors such as chance events or migration, so movement patterns. Now, the allelic frequency will again influence kind of the composition of the gene pool. Some alleles may be more more well represented or higher represented in a population than others, but that can change with different events in the environment. Typically, if the gene frequencies are not changing in a population, we say that that population is in what's called Hardy-Weinberg Equilibrium. And we'll talk about that in the next lecture about the Hardy-Weinberg Equilibrium and equation. But largely in nature, your populations are changing. You're having changes in gene frequencies. They may be kind of a smaller scale or a slightly more significant, but this is what explains how populations evolve, right? Changes in allele frequency within populations drive evolution. And again, that allele frequency is the frequency of a particular allele in the population. When we talk about phenotype or phenotypic frequency, this is the proportion of a population with a given phenotype. So how many individuals have coats with long hair, how many have short hair, or what are the differences in beak size, etc. Those are differences in phenotype. So they would represent the phenotype frequency. Genotype frequency is the proportion of a population with a given genotype. Genotype is the genetic composition of an individual, right? So if an individual was considered a heterozygote or a carrier for a specific gene, they would have both alleles or allelic versions of that gene. So they could be big A, little a. That's genotype. Again, their genetic composition. So your phenotype is going to be correlated to genotype as well as environmental conditions. Now, moving to microevolution itself. Again, microevolution is evolution on a small scale. It's actually on the smallest scale, hence the name, micro meaning small. So this is a generation to generation change in allele or genotypic frequencies within a population over time. And usually that element of time is occurring over the space of one generation. So very small changes that occur gradually within that population. Now, again, as I said before, these macroevolutionary changes themselves, kind of looking at them individually, won't create a new species. But the cumulative effects of microevolutionary change will eventually themselves be noticeable in phenotypic changes and therefore contribute to macroevolution. So at some point, cumulatively, again, they can contribute to the formation of a new species, but initially on the individual level, they cannot. But they can create subspecies, which are just variations of individuals within a population. They're not specifically new species yet because they can interbreed with one another, but they have differences in their genetic composition, variation. Now, there are 5 factors that drive microevolution. The main ones, or these ones being genetic drift, gene flow, mutation, natural selection, and non-random mating. Sorry, that one's a little difficult to see, but that's non-random mating, which is a type of sexual selection. Right? Now, these 5 drivers, per se, of microevolution contribute to changes, again, in the genetic composition of a population, right? And that's the basis of evolution. Evolution is the change in the genetic composition of a population. So these factors will modify allelic frequencies to change the gene pool, and therefore that changes the genetic composition of that population, and from there you have evolution. So these factors either act on or augment genetic variation within these populations. And we can, we'll dive into each one individually with kind of more depth of detail. But just giving you kind of figure out, stop, that's my USB. But giving you kind of a quick and dirty description of what each one is. Natural selection, you should, okay, here, you can have your, here, there you go, take your toy, go on. Natural selection, so this refers to or operates to increase the frequency of alleles that increase fitness. So if there is some sort of environmental change and there is a favorable phenotype that can deal with that sort of change, it will increase within the population. So the allelic frequency of that favorable phenotype and genotype will increase in the population due to the environmental pressures. Genetic drift refers to these random changes in allelic frequency. And they don't necessarily increase fitness. They can either increase or decrease fitness. But usually it runs into a bit of trouble because there's a loss of variation as far as your allelic frequencies due to some sudden environmental change or event that reduces the population. So usually what's happening is there's some chance event that causes only a small amount of individuals to kind of come out of the original population, and that creates a very small population size. When you have a small population size, you decrease the amount of variation within that population. It's no longer truly representative of the original population. So that loss of variation, that's a decrease in allelic frequencies. Gene flow is when you have the movement of individuals between two or more populations. So this would be immigration or emigration, right? And that changes variation, again, your allelic frequencies, because individuals are either bringing in new genetic material or genetic material is being pulled out of the populations. So you kind of have this ebb and flow of genetic material into and out of populations and between populations. That's gene flow. For your non-random mating, again, that's a type of sexual selection. This is when mate choice is no longer based on equal opportunity, right? So not every pair or mating pair has an equal chance of successful reproduction, right? Mating has become selective and based on some sort of characteristic. And then the last driver is mutation. Mutation refers to any permanent changes in the base sequence of DNA. And this typically serves as a continuous introduction of new alleles. And again, these new alleles may not necessarily benefit fitness, right? We have, as we'll talk about, either neutral mutations, some may be harmful and others may be beneficial. But these mutations introduce new alleles into the population, and those new alleles can then be established in the gene pool. Right, so that's a quick and dirty overview of the 5 drivers of microevolution. Again, we're going to go into depth with each one. But microevolution itself is highly observed in nature. We've observed numerous cases of it. And the reason why it's really easy to observe this is because of that short time scale of which that evolutionary change is occurring. So because it happens so quickly, and usually you can see these changes appearing in either the next or within several generations, we can directly observe these events. So a great example of kind of microevolution in action is pesticide resistance. Not only pesticide resistance, but herbicide resistance, insecticide resistance, and antibiotic resistance with your bacteria. They're all examples of microevolution, and all of them are more specifically microevolution occurring via natural selection. Right, so a big example of this was the insecticide resistance that was developed by some insects in Asia to the insecticide DDT. And that was an insecticide that was used intensively in these agricultural areas, And because of natural selection, because of random variation in these populations, there were some individuals that were resistant or could survive that exposure to that insecticide or pesticide. And therefore, that allele is going to increase in frequency within a population because those individuals are going to survive and be able to mate and reproduce and create fertile offspring. So as a result, you had the development of insects that were resistant to that chemical. Another big example, is with antibiotic resistance. This is causing big issues medically that is not getting enough attention, particularly the trouble with gram-negative antibiotic resistant strains of bacteria. But one that you hear a lot of, or is notorious kind of in this discussion, is the Enterococcus bacteria, which you find in the your GI tract. A lot of these bacterial species have evolved resistance to several kinds of antibiotics and several classes of antibiotics, which poses trouble when you have an infection and you need to be treated. Another example of microevolution in action is with our sparrows. House sparrows were originally introduced to North America back in 1852. And since that time, these sparrows have gone on to evolve different characteristics in different locations. And with the house sparrows, the kind of the most notable difference is body size. Right, so house sparrow populations in northern regions typically are larger bodied than sparrow populations that are more southern occurring. Now, this divergence in populations, at least partially, is likely a result of natural selection following Bergmann's law, which states that typically you find larger bodied organisms or species occupying colder climate areas because they are better adapted at dealing with the cold. They are better able to retain body heat than smaller species. So likely this is a reflection of just that. Your larger bodied birds can survive better in lower temperatures than the smaller bodied birds can. So likely the cold weather that's observed in northernmost areas or more northern regions, likely that's selecting for these larger bodied individuals. Song sparrows also are another great example of microevolution. Hey, do you want to not eat my shirt? Can you not eat my shirt? You have food in your food bowl. How about you go eat your food? Can I have my shirt back? Thank you. Dear Lord. Okay. I guess all the talk of birds is making me real hungry. So the song sparrow is another sparrow species that's found throughout North America. There's actually 31 subspecies of song sparrow that have been described. Now, along the Pacific West coast of North America, the subspecies of song sparrow form this cline. So a cline, again, is this gradual change or difference in characteristics and traits. They form this great cline in body size, in plumage coloration, and song characteristics. Those, again, all are examples of microevolution formation of your subspecies. All right, so let's actually go in depth or more nitty-gritty with the individual drivers of microevolution, right? These are the mechanisms by which microevolution can occur. So we'll start with genetic. So what is genetic drift? Well, say you have a healthy population, right? And some natural disaster occurs that causes a crash in population size, or a plummet in the size of that population. Because of that event, allele frequency is going to change, right? And this is genetic drift. Genetic drift is the change in gene frequencies or allelic frequencies within a population because of chance events that go on to create small population size, right? So there's kind of two reasons why it happens. First, there is some event that occurs, and second, that event reduces population size. Some individuals are able to survive, some do not. So because of that reduction in population size, you reduce allelic frequency, right? That random change is going to create these differences in allelic frequency. And again, that's due to these chance events. When we talk about genetic drift, what we often see occurring is what we call the random fixation of alleles in a population. You could also see the loss of heterozygosity, depending on which individuals survive, which ones don't, and the mating preferences of the surviving individuals. But when we talk about kind of the allelic composition of these populations, each population per se follows its own unique trajectory or unique evolutionary path. There's no uniform path that is kind of generalized across all populations. Again, each one is unique depending on the events encountered or the random changes that do occur. So when you have a situation of genetic drift, some alleles may become fixed in populations, and other alleles may not, or other alleles may become fixed in one population but not another. And when we talk about the fixation of alleles, this is referring to the tendency of allelic frequency to drift to 1, right? So if you reduce population size and the individuals that survive happen to be largely or all homozygous recessive, you're going to have fixation of that allele, the little a allele, because all individuals are carrying it. So again, fixation is talking about kind of the kind of like takeover of an allele within a population. It's increasing to a level where almost all individuals carry that allele. It's highly present. Again, this won't happen for all alleles. Some will move towards fixation, others will move towards elimination. So this is the disappearance of those alleles from that population. Now, fixation itself occurs more rapidly in smaller populations, and in larger populations, it occurs much slower. But eventually, no matter what the population size is, it does occur, right? So eventually, you will or you might have the fixation of certain alleles and elimination of other alleles within populations, regardless of their size. Now, when alleles become fixed, generally there's an overall decline in heterozygosity. And that's because each population is becoming more and more homozygous for one of the alleles. Right? It's either they're becoming more homozygous for the big A allele or more homozygous for the little a. But again, you start weeding out or eliminating heterozygosity, fixing homozygosity within the population. Some other trends that we see with genetic drift, as I have written up here. Genetic drift tends to decrease genetic variation within a population, right? Which makes sense because, again, genetic drift is occurring when you have some random event that significantly reduces population size, right? So usually where you can think of someone coming along and say they have really, really big feet, right? And they trod right over a little ant colony and say they take out like 75% of that original colony. Well, the 25% that are left, again, the genetic variation that exists within those individuals is random, but it's also going to be significantly less diverse or less representative of that original colony, right? As shown here, this event has left or only these three individuals. And those three individuals are all homozygous recessive, right? So that's going to go on to influence the next generation. That next generation is not well representative or well reflective of the original population. And that's because you have a decrease in that genetic variation because you're decreasing the amount of individuals that are representing that population. So while you have the decrease of genetic variation within a population, you do have the increase of variation between populations, which makes sense too, because as I said before, these new populations that are formed are generally significantly different or not well representative of the original population, right? These the allelic frequencies that are going to occur are going to be significantly different from that founding population, and therefore you have significant increase of genetic variation between those populations. Now, one thing to note, to make sure you don't get confused on, yes, genetic drift plays a major factor in evolution. Yes, it's a major factor causing evolution, especially when you have the division of populations. But the process itself does not involve natural selection, right? So the individuals that are surviving and going on to create the next generation after some chance event that has led to genetic drift, they are not surviving because of natural selection. They are not surviving because they have some adaptation that makes them better suited or better able to survive in these new environments. They simply exist because, well, in the case of our ants, those individuals didn't get squished, right? They are surviving by random chance, right? So everything with genetic drift is kind of this element of randomness. Random events, random reduction in population, random individuals left to form that new population or newly formed population that's now reduced. So again, these individuals that go on to survive, they're not surviving because they are better suited than the rest of the individuals. They are surviving merely by chance or by luck. And kind of going a little more in depth with something I said earlier, I mentioned that genetic drift is more significant with small populations, or it deals with the change of allelic frequencies within a population because of a small population size. Again, these chance alterations in allelic frequencies within a population, they're going to have the greatest impact on small populations, right? It occurs in all populations. It's felt within all populations regardless of their size, but the effect at which it's felt is strongest in small populations. Again, that doesn't mean it doesn't occur in small or large populations. It occurs in all populations, but again, the effect is strongest felt or more greatly felt in your smaller populations. And you may not instantly recognize why that is. So consider taking a small sample of individuals from a large population, right? So we have this giant little ice island of penguins. We're going to break it in half. We now have a significantly smaller sample of those individuals from that larger population. Now, if only 2 individuals, right, from this small population here were picked, they almost certainly wouldn't reflect the allelic frequency of the larger populations, right? And in some cases, they can't even do so, right? As seen here, right? Here's a really small population that's now been dramatically cut down. This frequency here is very different from the allelic frequency here because it doesn't even include your poor little red penguin. Maybe he got sunburned or something. But this penguin, or the allelic composition, the genetic composition of this penguin is not represented in this new reduced population. Right? The same will hold true if you pick 3, 4, or 5 individuals. As the selected sample gets larger, it becomes more likely that that sample you're dealing with will reflect the allelic frequency of the larger population because you're including more individuals, right? More individuals are randomly included. There's a higher probability that you're going to have at least one individual in there that represents some different variation in the population. So when we talk about, again, sample size and reflective of kind of the larger population, if you actually wanted to match, if you say you had a sample population, you wanted to exactly match those allelic frequencies and genotypic frequencies of the original population, that is quite quixotic because it takes a very large sample size, like in the thousands, to have a good chance of the sample, again, essentially matching. So when we talk about statistics, right, we talk about these using larger population. sizes, usually trying to use more than 30 individuals. Because as you increase the number of individuals used, it's more likely that you're going to have a sample that's more indicative or more representative of that larger original population. So because you have smaller populations, because there's less individuals in those populations and less variation in the genetics of that population, the change in the allele frequencies is going to be more rapid, right? So the allelic frequency, or the change of these allelic frequencies, is more rapid in smaller populations, and they're slower in your in larger populations, right? Because if you start off with a small population, again, that any sort of drastic change is going to have a significant significant impact on the genetic composition and result in huge losses of alleles. It can result in the loss of an entire allele. But if you have a larger population, there's more chance of diminishing the amount of change that's felt as far as the frequency of alleles within the population. So how do we get smaller population sizes? Well, there's many ways that this can occur, but the two general mechanisms that lead to small population sizes are one, genetic bottlenecks, and two, the founder effect. All right, so what the heck are those? Well, the bottleneck occurs when populations undergo periodic crashes. So you create a genetic bottleneck by dramatically reducing population size. And the allelic frequencies that will be present after that crash likely are going to be significantly different from those before the crash, right? So it's a change in allele frequency in a population due to chance following a sharp reduction in in population size. So looking at the bottles here, right? Here's your original population. Some event happens. Maybe there's an environmental change or a natural disaster, but that greatly reduces the size of a population. Right? So only some individuals from this population will go on to survive or form a new population. Now, the allelic frequencies of that surviving population, they're going to be different from those of the original population because some individuals will make it, some won't. And again, that has no correlation with natural selection. It's all by chance. So there is a chance that some alleles may not be represented, or some may be more represented in this new population than others. But whatever the case, this new population will go on to grow right after this bottleneck effect, and the gene pool or the genetic composition of this population will follow an independent trajectory that's different from the original population because it now has different allelic frequencies. So you get these two new populations, right? This new population that's been formed by the bottleneck is very different in regards to the genetic composition and allelic frequencies from the original. It's not well representative of the allelic frequencies from that original population. Now, usually, when we talk about these genetic bottlenecks, we see them a lot with endangered species, right? Endangered species can face genetic bottlenecks that are almost on a species-wide scale. And usually these populations or these species will suffer long-lasting effects that, again, are felt or burdened long after the population size later recovers. And that was a big thing that was seen with cheetah populations, which we'll talk about in a moment. But before we move into that, again, a bottleneck is a severe reduction in a population brought about by some sort of natural event or environmental change that shifts selection pressure, and it causes a reduction in population, and a new population is regenerated by only a few individuals, the few individuals that go on to survive. And the genetic diversity of that new population is greatly diminished compared to the original. All right, so moving into our cheetahs, right? Cheetahs, they've gone through quite a bit in their evolutionary history. Cheetahs survived 2 bottleneck events. The first one occurring during the last ice age event, which was about 10,000 years ago. They narrowly escaped extinction right at the end of that event. The second event is more recent, which was a dramatic bottleneck event that occurred in the 19th century, and that was due to poaching and habitat loss. When we look at cheetahs, all cheetahs share a small number of alleles. I have a picture of the bottleneck here. So this was the original population, right? The original allelic composition or genetic diversity of that larger population. You go through a bottleneck effect or event. Only these alleles will be go on to be representative of a new population, and this creates a smaller population with allelic frequencies, again, that are different from the original, also a lot less genetic diversity in this new population. So more specifically with cheetahs, their diversity level is less than 1%, right? So the genetic diversity of these cheetah populations is less than 1%. It's almost as if all cheetahs are identical twins. They are more genetically alike than inbred lab mice, which is incredible to think. And it's also incredible to think that these populations are still surviving even with that significant decrease or lack of genetic diversity. So why do they have such limited diversity? Well, it's because all modern cheetahs have arisen from, or it's thought that all cheetahs have arose from a single surviving litter, right? A single litter survived these bottleneck events. They went on to reproduce and form the modern populations that we have now. And that's what we believe accounts for this lack of diversity. And obviously, lack of diversity has significant impacts as far as population health and survivability of these populations. So the lack of genetic diversity in cheetahs has led to such things as sperm abnormalities, decreased female fertility or decreased fecundity. They have very low reproductive rates, high cub mortality, and immunosuppression. So these individuals are notorious for being immunocompromised, meaning that they are very sensitive to disease, very sensitive to any sort of environmental change that seems to put stress on their immune system. And as a result, they become more susceptible to disease. Now, when I was talking about the bottlenecks before, I said that there's you're going to have some sort of change over time. Those changes that are seen in this new population, they may, as the population grows, that population can be restored to kind of this normal genetic composition per se. That can happen in some instances, but for cheetahs it has not. Right. So since these bottleneck events, there's hasn't been enough time as far as the evolutionary time period for cheetahs for random mutations to accumulate within these populations and produce new genetic variation. And again, you have that element of insufficient time, but there's also the fact that because of their low genetic diversity, they have low reproductive rates. So there's these two major factors that are kind of compounding the fact that cheetahs aren't able to create enough random mutations, or enough random mutations aren't able to build up in cheetah populations to produce variation. Okay, so that's again all about our poor little cheetah friends and genetic bottlenecks, right? So genetic bottlenecks, as I said, were one of the kind of general mechanisms by which genetic drift occurs. The second one that I had mentioned is called the founder effect. The founder effect is a special case of genetic drift. In population genetics, the founder effect refers to the loss of genetic variation that results from or occurs when a new population is established by a very small number of individuals from a larger population. So small percentage of that original population moves out of the original population to form a new population, right? It's a new colonization event. So this small portion, right, of that larger population migrates to a new area, creates a new population. Now, when this new population is established, again, typically only a few individuals, which we call founders, are involved in colonizing that new area or serving in that colonization event. And we refer to this as essentially being an isolation bottleneck for the new population. Again, you're taking a small subset of the population that has moved out of the original, and only a few individuals are representing this new population. So the small size of a founder population almost guarantees, that its allele frequencies, one, will not be identical to the parent population, and two, are going to be significantly lower as far as genetic variation than the parent population. Again, that's because only a few number of individuals are forming this new population. And as I had said before when we were talking about our penguins, when you take a small number of individuals, right? Say we have a glass of different colored jelly beans, right? If I take 2 jelly beans from that jar of 1,000, it's very likely that those 2 individuals are not going to be well representative of the entire allelic. Well, we can't say that beans have an allelic composition, but we'll say that they're not going to be representative of the diversity that exists in that jar of jelly beans, right? You may be missing some colors, some flavors, maybe some textures, etc. So with this founder effect, you're going to have significantly lower allelic frequencies because you have only a few individuals that are forming a very small population. And those few individuals will go on to reproduce, and those events are going to either fix or eliminate certain alleles, with one allele usually becoming fixed in a population, the other being eliminated. This was seen with, or this still is seen with, Amish populations. So there are almost 250,000, I think, Amish in the US and Canada today. The present Amish population that we have today originally grew from 200 founders who came to the US from Germany and Switzerland in the mid 1700s. Now, since that time, Amish have followed a very simple lifestyle. For example, they do not own cars. They instead travel by horse and buggy. They do not use electricity. Arts are limited. But the major thing that deals with the genetic diversity is that they rarely intermarry with people outside of the Amish population. So reproduction solely happens within your Amish gene pool or population, which means there is no variation that's being brought in through immigration or migration events. It also means that there are cases of interbreeding because these individuals are so closely related. One of the original 200 Amish founders carried a recessive allele for a rare condition called Ellis von Creveld syndrome, which is a type of dwarfism. Today, the Amish population has more cases of this syndrome than any other population, any given population in the entire world. And it's because these individuals do not intermarry outside of the Amish community. And it's also because the original population that these cultures have stemmed from was a very small founding community. Again, only 200 individuals. But because one of those individuals had this allele, it became highly present in the population. Again, because there's no intermarrying that's occurring. Ellis-Revolt syndrome. Again, it's a type of dwarfism. It's a rare genetic disorder. It's autosomal recessive. It's known as a ciliopathy. So ciliopathies are genetic disorders dealing with either mutation of the cellular cilia, their anchoring structures, basal bodies, or the function of the cilia themselves. But the syndrome itself is characterized by short-limb dwarfism. You can have polydactyl, meaning there's additional fingers and or toes, abnormal development of fingernails, congenital heart defects. This occurs in over half of the patients, predominantly atrial septal defects. But there's also intellectual disabilities, renal abnormalities, changes in the dental structure, and abnormalities of the teeth and mouth themselves. One of which is a shortened upper lip. That's kind of connected to some of the nose structures, and as well as the alveolar ridge and neonatal teeth. But again, this is an interesting case of a genetic disorder that is now extremely present in a population because of this example of founder's effect, right? A small subset of an original population moved out of that original population, established a new population, and that new population was created by a small number of individuals. One of those individuals happened to carry this recessive allele for this condition, and because of their cultural practices, the fact that you had these kind of pre-existing influences or factors as far as small population size, this disorder has been able to become highly present and nearing towards fixation in the population. What? Go play with your toy. Yes, go play with your toy. My computer's not the toy. No. All right, so moving on to another factor, right? Our second factor. So that was all genetic drift. One of the other drivers of microevolution are mutations, right? So mutations refer to any permanent alteration in the makeup of DNA that is heritable. Usually these are alterations that remain fixed in the genome or will occur because of errors in DNA replication or errors in DNA repair. Because they do not get repaired, they become fixed in the genome. And once they're fixed, they can become inherited. Usually, when we talk about mutations, we're talking about base pair substitutions, deletion, translocations, etc. In general, mutations increase variation within the gene pool of a population. And really, mutations serve as kind of the ultimate source of genetic variation. A lot of the genetic variation that's needed for evolution is produced through mutation. But it's important to keep in mind, again, mutation isn't occurring to benefit a species per se. Remember that mutations can either be neutral, some are harmful, they can also be beneficial. So again, they're not themselves working directly to further survival and reproduction. That comes as a byproduct of their influence on functioning. Harmful mutations themselves are not likely to account for these. What was that? That's my cell phone. That's my cell phone. Go play with your toy. Go play with your toy. No, my hand is not your toy. Oh. Look at it. Dear Lord, I feel like a mother. Okay. What was I talking about? Harmful mutations. Right. Okay. So harmful mutations themselves are not likely to account for significant changes in allelic frequency. These changes are more likely to be brought about by good mutations or beneficial mutations. Again, the good mutations likely will bring in new genetic information to the genetic pool. It's going to benefit those individuals in some aspect, and because of that favored impact, it's more likely that mutation will, or the allelic frequency of that mutation will increase in the population. But again, whether they're neutral, harmful, beneficial, whatever, their mutations themselves are necessary to increase the variation in a population, so that natural selection itself can occur and produce organisms that are more suited to their environment and create kind of the foundation upon which evolution can occur. All right, so let's move on to the third driver of microevolution, which is gene flow. So gene flow is a change in allele frequency that, or deals with a change in allele frequency that occurs when interpopulation mating occurs. So what? So interpopulation mating means that members from one population are mating with the members of another population. And because you have kind of this cross-population mating, or your interpopulation mating, there's this constant movement of genes and alleles from one population to another. And again, the movement of alleles is between different populations, but both populations are of the same species. Now, gene flow itself can occur through various mechanisms, right? When we talk about gene flow, again, we're talking about the movement of genetic material from one place to another. In this case, from one population to another. That's gene flow. When it occurs, or how it occurs, is through migration and or dispersal. Right? So migration deals with the movement, the physical movement of individuals. Dispersal is more of the movement mechanism that you see with plants as far as dispersal of seeds or spores. So let's talk about Figaro. Stop playing with the curtains. What are you, a 5-year-old? Actually, you are 5. Okay, I guess you can act like that. So migration. Migration is when individuals move either into or out of a population. So in an evolutionary sense, we're talking about this as far as microevolution. Migration deals with the movement of alleles between populations. Right, so now naturally alleles don't move by themselves. They'll move as organisms disperse from one population to another population, or they move between populations. Now, when we talk about migration, there's immigration and emigration. Immigration is the movement of individuals into a population. And as individuals move into a population, they carry their alleles with them, and that introduces new alleles to that population that they're entering. Right? So alleles enter a population when new individuals move into another population. They bring their alleles with them. Right? That's, again, gene flow. Emigration refers to the movement of individuals out of population. Right? So alleles are lost from population when individuals emigrate or when they leave. Now, gene flow itself tends to decrease genetic variation between populations. The reason for such is because, again, you have this movement of individuals between populations. So this is causing alleles from one population to be introduced to another population, or the diminishing of certain alleles within a population, and the expansion or augmentation of other alleles in a population. But because you have this transfer of genetic material between the two populations, you're transferring similar or the same alleles. So because of that, the allelic frequencies or the allelic compositions of those two populations are going to become more and more similar. So again, gene flow tends to decrease genetic variation between populations. And when we talk about migration itself, please don't confuse the migration that I'm talking about in this sense with seasonal migration that you see with birds or the monarch butterflies. I'm not talking about seasonal movement patterns. I'm talking about, or the migration patterns that I'm talking about are either continual movement between one or two populations, random movement due to availability that's now made. In this example, there's a pass that's made in this mountain range, but it's not referring to these seasonal expeditions that are made by certain organisms. They're not truly moving from one population to another population and therefore transporting alleles. So that is kind of the bulk of gene flow. Again, a large amount of it is due to migration and dispersal. Migration, you can think of more as tailoring to your animals. Dispersal is more plant related. Again, as far as the dispersal of seeds or spores, that's going to influence again your plant populations, because certain pollen spores will land or will be transferred from this population of angiosperms to a new population of angiosperms. Angiosperms are your flowering plants. That operates as gene flow because pollen represents the sperm cells of plants. So again, that's gene flow. The fourth driver, we'll call it, is non-random mating. And as I said previously, non-random mating is a type of sexual selection. Right? So when we look at a population and we look at their mating probabilities, when the probability is the same, meaning that individuals are just as likely to mate with distant relatives as they are with close relatives, or with this individual versus this individual. There's kind of equal opportunity of mating potential. That is random mating. Non-random mating is quite opposite. Non-random mating occurs when the probability that two individuals in a population will mate is not the same for all possible pairs of individuals. So mating for one member or for a given member of a population is not equal to the probability or mating potential or mating probability as another given member. And this is usually, or this is mating that's based on either phenotype or female choice. Usually it's both. Females are choosing males based on a certain phenotype or a preference for a certain phenotype. So the male that's chosen based on the choices of the females and those preferences, whatever male is chosen will increase the proportion of his alleles in the next generation, right? So the alleles that, or the allelic frequency within a population is governed by these mating choices, with some allele frequencies having a higher percentage in the population. Those alleles coming from males that have certain phenotypes that make them more attractive as mate choices. So we can break non-random mating down into two kind of different classes, right? There's assortative mating and disassortative mating. Disassortative mating is not quite as common as assortative. This is when individuals only mate with others who are phenotypically different from themselves for a selective trait, right? So they select for their opposite, per se. Generally, this will lead to a decrease in homozygous genotypes for the genes that govern the selected phenotype, and also for any of the loci that are closely linked to those genes. Assortative mating, again, this one's more common. This is when individuals mate with others who are like themselves phenotypically for a selected trait, right? So they mate with those that are similar to them. So again, your mates are being sexually selected based on phenotypes. More specifically, their phenotypes are similar to the individual that's seeking a mate. So consequently, you have mating between individuals of similar genotypes. This can either be by choice, it's usually by choice, but it can also be due to environmental factors such as location. Now, the most common case of your assortative mating involves interbreeding, right? So this is mating events or sexual reproduction between close relatives. Kind of the most extreme case of inbreeding is self-fertilization, which you see with plants. So most of your angiosperms contain both male and female reproductive structures. So they are able to produce both male and female gametes. And as a result, plants can self-fertilize, right? They can use the pollen that they produce to fertilize the eggs in their ovary. That's, again, the extreme case of inbreeding. Now, inbreeding itself does not change allele frequencies, but it will increase the frequency of homozygous genotypes. So it's not, I should better word that, it's not changing the allelic types in a population. It is selecting for your homozygous genotypes. So the frequency of the homozygous genotypes will increase, but it's not necessarily selecting for one allele over another. Again, it's more influential. It's not necessarily influencing allelic frequency. It's more so increasing or influencing genotypic frequency. So mating, again, mating between these specific genotypes, that's going to change your genotypic frequencies. So that's pretty much non-random mating. Again, it's governed by female choice and the possession of certain phenotypes, or the characterization of certain phenotypes. The fifth and last driver of microevolution is natural selection, which you should know very well at this point if you're in my evolution class. Natural selection changes allele frequencies in a way that leads to adaptation in the environment, right? Natural selection selects for traits or selects on traits that are advantageous for survival based on certain environmental pressures that are felt at that moment in time. So populations that are undergoing natural selection, they are evolving. And they're evolving because you're having shifts in allelic frequency, right? Alleles that contribute to better fitness or greater fitness will increase in frequency over successive generations due to natural selection. When we talk about fitness, remember, fitness is the ability of an organism to survive, reproduce, and pass its genetics on to successive generations, right? So these surviving individuals, or individuals that are suited for certain environments, will have a higher fitness. They're going to be able to survive and reproduce, and therefore pass their alleles on to the next generation. And going, kind of taking another step back, as I said before, natural selection, it's going to select for traits that are advantageous for survival based on the environmental pressures that are felt at that moment in time. It's not static, right? So natural selection will change. It only operates based on the current environment. As conditions change, different alleles will be selected for. So when either the homozygous dominant or recessive is more suited to an environment than the heterozygote, then those homozygous genotypes will be more likely to become fixed in the population. But when heterozygous genotypes have advantageous opportunities over either one of the homozygous genotypes, then variation will tend to increase in the population for that heterozygous genotype. Right? You see that with what we were talking, the heterozygote advantage with carriers of sickle cell anemia, and they're by having such, it increases their resistance to malaria. Now, for natural selection to occur, you need to ask yourself, what does natural selection need in order to operate? What must exist for this to occur? I'll give you a moment to think. You can listen to Figaro cleaning his toes. The answer is genetic variation. Right? Genetic variation must exist for natural selection to occur. And again, as I said previously, the ultimate source of genetic variation is mutations. Now, once variation exists, it can be affected by independent assortment and genetic recombination during gametic formation, which occurs in meiosis. We had talked in class about the biological importance of meiosis, one being that it produces or forms gametes, but it's also a major source from which genetic variation can arise from. And that has to do with the effects of independent assortment and genetic recombination. So consider if we did a cross, right? We're going to do a dihybrid cross between two individuals. Both individuals are heterozygotes for gene A and gene B, right? So each has a genotype of big A, big B, little b. Now, if you do the cross with those two individuals, you get 9 different genotypes that arise. And that frequency arises from only 2 alleles at 2 loci, assuming that these genes are not linked. Now, what if there were 6 alleles possible, right? If there were 6 alleles possible, you could have over 4 million genotypes made. So there are thousands of genes in an organism, right? Each gene has different alleles, and there are many alleles that are possible at any one of those loci. So again, that's another huge source of great genetic variability in nature, right? So it becomes clear that in nature, your 2 sources of genetic variability are largely arising from the kind of random allelic combinations that can occur because of independent assortment and genetic recombination during meiosis, as well as mutation events. All right, so that covers everything for, everything I have to tell you about microevolution, as far as what it is and how it operates, or what mechanisms it operates through. If you have any questions, if you're in my class, you can ask me in class. Otherwise, you can feel free to put your comments in the comments section. And I hope, as I say with all my other lectures, that this either helped you to better understand the topic, or you at least are walking away with, if not a better understanding, something new that you have learned.